So today I'd like to talk about a new technology platform that our lab has developed over the last 5 or 6 years, and that is a delivery of mRNA through a carrier, mainly through extracellular vesicles for glioblastoma therapy. These are the objectives: to describe extracellular vesicles as a delivery system and to illustrate the potential for using mRNA therapy for brain tumor. And I have no disclosures. As you know, with the recent sort of COVID vaccine development, mRNA therapeutics has really been in the limelight. And there's been, a lot of great technical advantages of using mRNA therapeutics. Unfortunately, currently, the main formulation in which the carrier is given is through this ionized lipid nanoparticle formulation. This is a nanoparticle that's foreign material that is given, and because it's foreign, majority of these materials get stuck. More than 95% or so, depending on who you read, get stuck in the liver. So unless you're really interested in targeting liver cancers or liver-related diseases, lipid nanoparticles really are not that effective. These materials also do not bypass our physiological barriers, such as the blood-brain barrier. There are some immune reactions because these are foreign materials where it could be local or even systemic in rare cases. So we believe that the mRNA therapies that are available, really awesome, but it's just as good as if your carrier is good enough to carry them to the specific sites of interest. So just as a brief overview of what mRNA therapies and how they work, is that you have mRNA that are packed in these carriers, mainly lipid nanoparticles. And essentially, you have these sort of cells that then take these nanoparticles in through the processes of endocytosis or phagocytosis. And these mRNA that are encoded for a particular protein that you'd like for them to be translated into, are then they enter these cells. The cells then, and mainly in the case of COVID vaccine, white blood cells that recognize and they start producing proteins that these mRNA are encoded for, and then they eventually create antibodies and to generate these antibodies against the virus. And so currently there are many carriers that one can use that go beyond just mRNA as the cargo, and that is the cellular-based, like dendritic cells or extracellular vesicles that we'll talk about today. There are also carriers of, you can also use peptide as a carrier, viruses, and different nucleic acid formulations as well. And so here we have a schematic of the different cargoes that you can use to deliver the agent that you're interested in. And that, as I mentioned, cellular base, viral base, peptide base even, and the mRNA base that we're going to talk about today. So the mRNA technology that we've developed, and when we compare directly with some of these carriers, is that there are a lot of limitations, as I mentioned: targeted delivery, inability to go beyond the liver, immunogenicity, and I think all of these issues can be rectified with the use of exosomes, mainly because we're able to generate the mRNA endogenously. So using the toolkits that your body already has, we're able to then transcribe the plasmids that we're going to deliver into mRNA of interest. So the story that I'm going to share with you are two projects or studies that have been published recently. And that is that the extracellular vesicles. And what are they? Well, every cell, they're able to communicate with other cells that are not directly nearby by shedding little vesicles that are made constantly throughout its lifespan. And within these tiny little vesicles, they're sort of loaded with cargo information that's going to sort of give to another cell that's distant or further away, whether it be immune cells, whether it be similar cells at another sort of location, where they have nucleic acids, for example, heat shock proteins, and other metabolites that are in there that are going to sort of message to other cells. It also is decorated with certain biomarkers or markers that are specific for those exosomes. Mainly, it has some markers that are, similar to the source of where this cell is coming from, but also additional markers that are specific for that exosome. They're tiny. These exosomes can range from 40 to 50 nanometers to 1,000 nanometers. The difficulty and the challenge as to why we don't use exosomes routinely as cargo are these three items. One is that it's really difficult to insert large cargo. So if you're wanting to load and package these, once you are able to isolate these extracellular vesicles, to be able to stuff them now with these large sort of mRNA is just technically very challenging. The generation of these exosomes, as I mentioned, cells just create these exosomes throughout their lifespan. So if you want to really accelerate the production of EVs, then you need to have a more efficient way, because it's going to, cost you a lot of money, and also it's going to take a lot of time to generate them. The ability to also modify the surface for targeting purposes, because, if you want to ensure that that exosome specifically goes to another cell of interest that's further away, then you need to be able to surface modify them so that it can target. So as I mentioned, all of the limitations of the current lipid nanoparticle formulation, I think, can be bypassed by this technology. And so the first story that we were able to show that large-scale generation of these was through a technology that we developed. It's called cellular nanoporation, or CNP, as we're going to call it. And so this is a schematic of the platform that we developed. And that is that we have a chamber system and a little microchip that has little holes in them, these nano sort of size channels. And in this pool of fluid, we also have a plasmid of interest. And that plasmid will then eventually become the protein of interest. And so here for brain tumors, we place plasmids for P10. So this is a system that if there's a tumor suppressor gene, so tumor genes that are not expressed, we can replete almost any gene of interest. So as a proof of concept, we use P10. We also use P53, and eventually we were able to show that you can actually put a cocktail of plasmids, so not just replete one type of protein, but multiple proteins. So that's also a cool technology. And what we do is we place a source cell, and here for this paper we used embryonic fibroblasts, but depending on the cell that you're interested in targeting, we plate these cells, and we've sort of optimized this electrical system where there's low, gentle energy that flows through this pool of fluid. And at the point of contact where the source cells are, the plasma membrane opens transiently. And during that sort of opening of the plasma membrane, the plasmids in solution enter these cells. And for mechanisms that were unclear at this moment, the electrical energy stresses the cells, we think, and the extracellular vesicle formation increases by about 50% more. And the encapsulation of plasmid into mRNA is like more than 1,000-fold more efficient. So what we have, here in a system where you have these cells that are stressed under electrical energy, extracellular vesicles are formed in a very highly efficient manner. And during this process, we're able to pack small plasmids that then eventually become the mRNA that we're interested in. So we have plasmids for P10 that we put in this fluid, then translate into mRNA for P10, and your body then produces the protein for P10. And so what we do is that once we have these extracellular vesicles with the mRNA, we then isolate them, and we then deliver it to the area that we're trying to deliver, whether it be IV or local. So here's another schematic of what this looks like with the source cells, electrical energy, where the plasmid of interest is going in there. And what happens in terms of extracellular vesicle formation, it actually enters these multivesicular body. And within these multivesicular body, tiny exosomes are formed within it, intracellular bodies are calling it. And then eventually it gets sort of shuttled out of these cells that are under stress, and you've got this extracellular vesicles or exosomes that are formed. And they're packed with these mRNA now, and these will then travel to different parts of our body, whether it be for the brain or for, we have breast cancer model, metastases model, where it'll then home to these specific cancer cells of interest, and then it will be endocytosed into those cells, and the mRNA will be delivered to, let's say, the GBM cell, the tumor cell, and the mRNA for P10 then gets translated into P10, repleting that gene. And so during that thermal sort of local thermal sort of energy through the electrical forces, we see that there's a generation of the heat shock protein with the cellular nanoporation in the Western blot here as compared to our control. We also see, increased sort of upregulation of some of the tumor suppressor genes, P53, and also TSAP as well. And then we also see significant increase in the extracellular vesicle biogenesis. This is just a thermal sort of study that I won't go through here. And this is electronic microscopy, a photo image of the multi-vesicle body, or MVV as they're called, where, when you give that electrical energy, significant sort of intracellular bodies are formed that eventually are then shuttled out of that cell and become extracellular vesicles, if you will. And you can see that our cellular nanoporation generates significant many-folds increase in the multivesicular formation, which is the biogenesis of a lot of these exosomes. And we also show that when we look at these longitudinal studies, we essentially make, if we make these exosomes to fluoresce in the red regime. And so these are, the red sort of signifies the exosomes. And over time, exosomes enter the source cells, as you can see in these big sort of red blobs here. But over time, in about 1 to 2 minutes, that entry stops, so telling us that the plasma membrane sort of opens transiently and then it closes back. So we're not, injuring the cell per se, where the cell is, unhealthy and they're dying. It's just a very transient, very gentle way of transiently opening up the plasma membrane to allow us to deliver the gene product. What we show here is that when we use the exosome and we've targeted specifically for glioblastoma through decorating the surface of the plasma membrane or the membranes of the exosomes with EGFR variant 3, we're able to show that our targeted therapy, if we're able to inject it intravenously, the exosomes enter the brain, bypassing the blood-brain barrier and entering the brain. And you can see after 4 hours, it's still, it's starting to accumulate, as compared to, let's say, the liposome. Majority don't end up there because they're probably all getting cleared and stuck in the liver. And this is exosomes where you'd see a little bit there, but it's not targeted specifically to the brain. So it's probably still getting cleared in some capacity or not getting to the brain per se. This is just a tumor model demonstrating that we're able to target and be able to sort of, show long-lasting sort of residing, the exosomes sort of stay there for up to about 8 hours or so. This is just a in vivo study demonstrating that in an animal model, and this is two models. This is a syngeneic glioblastoma-like model, the GL261 tumors, in an immune competent animal, but also in an immune incompetent, the skin mice that have human cell line of GBM. We show that when we inject these exosomes that are targeted in these tumor models by tail vein injection, that we see exosomes significantly accumulating in the tumor as compared to our controls, and that includes liposomes that we used with the same cargo. And then we also show here, similarly in the U87, significantly increased in the exosome spatial distribution along the brain tumor. When we look at the different organs, we show that, this is lipid nanoparticles. Majority of it just gets stuck in the reticuloendothelial system, the liver and the spleen, which is what we would expect. The exosomes that are not targeted still get to the brain because that's what they naturally home to, but some still get stuck in the liver. And here with the targeted exosomes that we have, where we surface modify the exosomes, significantly increased numbers go to the brain for both models of this GL261 in the U87 models. This is just to show the survival benefit and also the tumor control. Basically, we were able to show that there's improved survival in these animals, and we now have additional unpublished data that when we do combination therapy, that we can actually get or achieve a flat line where there are long-term survivals in these very aggressive GBM models, where about approximately 40 or 50% of these animals survive long-term. And I think what we're doing is really changing the local tumor microenvironment milieu through the combination therapy, which has not been published yet. Just to note that XOT just means targeted exosome. This is the same exosome without the mRNA cargo. So it's just basically a targeted empty exosome, just to demonstrate that you need the cargo, the mRNA, to do its job. This is to demonstrate that we have, in fact, restored a tumor suppressor gene, P10, as you can see in the Western blot here, that we've completely restored it. And that there's significantly increased, at least 50-fold increased in the expression of P10. You can see in these immunohistochemical stains here as well that these brown sort of spots that you see increased P10 expression as compared to the empty exosome in our controls. And what I wanted to do here is just continue. So, 3-4 years later, we then developed the next generation. And that is, can we actually create even more exosomes that's going to be produced at a high throughput level? Because ultimately, we want to be able to create this for patient use. And can we create a model system where the exosomes that are produced and adapters on the surface of these exosomes, so that all you need to do is really do a plug and play approach where you can just, place an antibody or a targeting agent of interest. It's protein based and it'll just stick to that receptor. And so this was just recently published a couple months ago, several months ago now. And basically the schematic here shows is that rather than using a stationary microchip, like a chip device, we've created a microfluidic device where the source cells essentially are not stationary, but they flow slowly through this microfluidic device. And as they're flowing at the point of contact, we have electrical energy that goes through these cells, and during that time, this whole system is in a pool of the same fluid with plasmid, and essentially the plasmid is delivered into these cells, creating the mRNA-loaded extracellular vesicles. And it's just a schematic of what happens here. And what's really cool about this technology beyond the microfluidics part is that there's an adapter, CD64, which essentially, we've been able to decorate onto the plasma membrane of the source cells. And what happens is that pretty much any antibody, monoclonal antibody that we use clinically can be conjugated very easily, very much a plug and play approach. You don't need a lot of fancy chemistry or conjugation methods. And so this is sort of a shelf, you can just take it out of the shelf and, place an antibody or a mixture of antibody that you're interested in and target that exosome to the site of interest. And in this study, what we did here is, again, we took several glioblastoma models. And this is, I believe, it's a GL261, but we have several other ones, CT2A and the U87, the SCID model, demonstrating that we're calling this immunoexosome because of the adapter that we placed there, that we're able to get these exosomes significantly higher through a systemic delivery method into the brain compared to all of our other controls. And that we're able to show that there's a significant tumor control and also survival benefit with just the exosome alone. And again, as I mentioned, with additional combination therapy that we've currently optimizing, but it's showing very promising results of long-term survivals in these animals as well, and some MRI images here demonstrating the tumor targeting, but also tumor shrinking over time. So, I hope I was able to convince everybody here that we are in fact able to create large quantities of exosomes at a highly efficient manner. But importantly, we're able to insert, large gene products through mRNA loading through this technology. And in fact, we're able to restore some of the tumor suppressor genes in GBM, as well as in other tumor types. We're able to show that this readily crosses the blood-brain barrier. We actually see that when we look at this in longitudinal, real life imaging with multiphoton imaging as well. So we see these exosomes crossing the blood-brain barrier with minimal to no adverse reaction, because it's not an immune foreign agent. And then our more recent sort of technology was able to adapt and adopt the initial version 1 and be able to add these more plug and play approach of putting tumor targeting agents. And it is currently limited by the fact that you need to use a monoclonal antibody to target. But I think that currently the state-of-art technology for mRNA therapeutics is at the lipid nanoparticles, but I think the next stage would be the cellular based mRNA therapies. And hopefully in about 10 years, it'll be a different type of a carrier where you don't need cellular machinery, because that is quite expensive, and getting it through the regulatory process for patient care is difficult. And so this is for GBM, but we have other studies that have shown that we can actually deliver collagen, for example, in fibroblast, and create this system for anti-wrinkling effect, where we wrinkle animals over a 3-4 week period with UV light, and they're completely wrinkly, and you place the exosomes that have collagen now loaded into the exosome, the mRNA, and the skin is completely flat and they're buoyant and anti-wrinkly. And so with that, we ended up then expanding that idea to maybe looking at anti-aging for the body, and even neuroprotection. And so there's and something, an area that we thought maybe could bring this quickly into the patient was looking at rare genetic diseases where there are genes that are so large that you can't actually use gene therapy because you can't deliver it. And so we're looking at those disease types as well. And I can stop there. So I should thank my lab. So this is my lab, members who have been working on the projects that I've described here, and some of our collaborators, and then some of our funding sources. And so I'll take any questions that the team may have. Thank you very much for listening.
That's really exciting, Betty. Thank you so much for coming to give a talk for us. I actually have a question, and well, first commentary. I guess if you make an anti-aging molecule, you'll be able to fund the rest of your research forever.
Yes, what's interesting is that I have 4 high impact, I think, like Nature series paper on tumors. I have 1 like Nature series on collagen, and yet I've been called like 12 times by pharmaceutical companies for the anti-aging, the anti-wrinkles. So I think you're right. I think perhaps one can sort of feed cancer research to save lives rather than.
It's sad that our priorities for brain tumor are like 0.001% of that for anti-aging. But I do have a kind of a conceptual question. You may have said it and I may have missed it, but basically you have a cell, you add a plasmid which has DNA, it gets transcribed into RNA, gene of interest, and then the electrical field buds off with a piece of membrane. But what was it that allowed that membrane to be specifically able to target CNS? You said, was it EGFR V3 was on it, or what else do you put on there to make it not go to the liver and go to the brain?
So, we make the source cell. So for example, before we actually do all the electrical pulsing, we modify the surface of that source cell so that we have them overexpress EGFR. or overexpress EGFR variant 3. So when the small membranes butt off, it actually has EGFR variant 3 on its surface, for example, or so that's how we sort of indirectly have it target. Now, if we had, and we can engineer them so that it'll eventually, and there's some markers that I don't talk about here, where it can sort of, we can help it to bypass certain organs so that it actually will go to the brain, or it won't go specifically to the liver. So there are ways to sort of engineer that source cell before the cargo is packed into the exosomes through the electrical energy.
Well, that's fascinating and very innovative technology. I thank you so much. Ben, are there any, I thought I saw a couple of questions on the chat, but I don't know how to pull that up here.
Yeah, so it says, Dr. Kim, so this is from Ken Klecki. Have you tested various electrical stimulation protocols, such as AC/DC, sinusoidal versus pulse? So full disclosure, I'm not an electrical engineering person, but these are really small, like pulses, like millisecond to nanosecond electrical pulses, and I don't know if that. I don't think that the AC/DC. I think that's like larger pulses. So the answer is I don't think we did, because we really needed like very small, gentle, quick sort of pulses that will not kill the cell and will not make them unhappy. They will still be happy enough that they'll, continue to produce these exosomes.
Great. Well, thank you. Suzanne, I know you're not supposed to go until a little 11:50, but you look like you're ready to go. So